numbers decline, and they're not able to modulate the cytotoxic T8 activity. Okay, so that's been shown to be another reason why autoimmune disorders may exist, because regulatory T cell numbers have diminished or declined. All right, so there are a couple of reasons why for that, and that's another one right there. Suppressor T cells or regulatory T cells numbers diminish, and they're not able to modulate or keep in check the cytotoxic T8, so they don't become overzealous and get a little crazy. Okay, does that make sense? So far, so good? All right. There's our perforant and granzymes there. So can you separate in your mind the antibody-mediated versus the cell-mediated, what they're going after? The cells are involved here. Even though we haven't got to antibodies yet, we'll come to those in a little bit. But you can separate what the T4 and cytotoxic T8s are associated with. There is a pretty clear demarcation there, and you want to be able to do that. All right, and I'll come to that in a second. So before this, let me slow down a smidgen here and just kind of say, what if, again, you take out that cell that produces interleukin-2, and we called that what? What cell produces interleukin-2? Again, without this particular cell, you don't have communication between cell-mediated and antibody-mediated. So now your immune system has been shut down. So what cell produces that again? Let me holler it out. T4 helper cells. Okay. Now there is a virus that has a propensity for destroying T4 helper cells, and if that's the case, okay. Now it's not the only cell that this virus infects. It also can infect macrophages. It can even infect microglia. What are those cells? Microglia. They're in the CNS. Yeah, they can also infect those too. And so we have this T4 helper cell, and we take it, H-E-L-P-E-R, helper cell, okay, and we get rid of it, okay. Now, what happens? No more interleukin-2, no more proliferation or differentiation of T or B cells, because this product, okay, interleukin-2, that cytokine is necessary for B and T cell proliferation, whichever one you want, B and T, proliferation. Okay, so now we have a virus that has a propensity for destroying and infecting these cells and reducing their number. What is this virus? HIV, yes, human immunodeficiency virus. That's a virus that has a propensity for destroying our T4 helper cells. So, if that's the case, now our viral load, if you want, is up, and our T4 cell number count, T4 count is down. Okay? There are certain values, and you can study these in the clinical course, that says, hey, if your viral load is this many, okay, and your T4 cell count is this many, okay, you then end up with this. Okay? That's your diagnosis because of your values of viral load versus T4 cell count. Okay? So, the question then is, what's the big deal about that? Now your immune system is for nothing. So what kind of infections might you get? There's a word for this one, yeah? What kind of infections? I know you said it. How many said it?
Saying everything.
Okay, that's great. But you know, what kind of infections do you get because of the availability or the ease of which these pathogens can live in this environment? They take the opportunity to proliferate. So you end up with opportunistic infections. Opportunistic. Okay? That's actually a word you want to attribute to that. Opportunistic infections. And there are actually in very specific illnesses and infections here that if you have it, there's absolutely no question that you have AIDS. Okay, absolutely no question. And the fancy word for this, and I won't write on the board, but it's called this pathognomonic. I don't know if it's a weird word, but it's pathognomonic. If you have this disease, there is no question on the planet Earth that would be, any other answer is you have AIDS. There's nothing else it can be. And there are certain pneumonias. Pneumocystis carinii pneumonia, PCP, is a type of pneumonia that if you have it, there's absolutely no question that you have AIDS. And that PCP is not what you think it is, I guess. But pneumocystis carinii pneumonia is that. That's a pneumonia that, without question, if you've got it, you have AIDS, no doubt about it. And these are opportunistic infections that normally we all can come back without any trouble. When your immune system was shut down, okay, because in fact you take out that T4, it's kind of like the town crier. It says, hey, I know there is an infection, but now I can't do anything about it. There's no one here to make the call. There's nothing here. They're the middleman. Take out that middle person, there's nothing to fire up the immune system, whether it be T cell or whether it be antibody. There's nobody there. So you have opportunistic infections that people die from. They don't die from AIDS. They die from opportunistic infections. And that's an example of one right there. Kaposi sarcoma is another one that is that too. SAR, sarcoma, okay? Kaposi sarcoma is another one. But these are, you can write these down. They're not testable, but let's do know there are disease infections here that are pathognomonic that says if you got it, there's no doubt, okay, and they all fall in this category of opportunistic. So that's my point right here is, yes, that is the case, opportunistic, and you die from opportunistic infections. You don't die from AIDS, but there are indeed infections that are notoriously seen with AIDS, and that's without a doubt. Pathognomonic is the word there. You don't have to jot that down, but there are indeed infections that if a person has them, there's no question that they have that disorder, okay? But opportunistic, that's a word you want to circle and recognize because that goes with this, okay? Does that make sense? Okay, some key words there with HIV has propensity for destroying T4 helper cells. T4 cells seem to serve as the town crier of sorts, okay? Because they are able to modulate and control both cell-mediated and antibody-mediated, okay? And opportunistic infections is the kind of the end point of that discussion, but people die not from AIDS, but from infections. Okay? All right. What can we say about this? I've talked a lot about this already. What about this? T4 cells. What are they attracted to? What MHC class are they attracted to? Two, okay. What is their function? T4 helper cells?
Cause proliferation.
That's exactly right. Cause proliferation differentiation of like the B cells, if you want, a good example there, by using interleukin 2. Okay? They are the ones being presented to by, whoever my list one, they are the ones being presented to, okay? By our APCs or B cells, macrophages, okay? All right. T cells, what are they attracted to? What class? Yes, class 1, and then what kind of antigen would they be? Would they be also endogenous, right? We could say that too. Endogenous antigens, something that's produced within the cell, like an abnormal protein from a viral infected cell. Okay? What is T8 cell? What is their function? They, what is it? To kill. Yes, they kill individual cells by doing what? Using perforins and granzymes, and they also perform that big long word that they're constantly scanning for abnormal cells or infected cells called immunosurveillance. Immunosurveillance. Okay? Alright, so B cells, okay? What are they going after? B cells are going after things that are in the fluid, yes. Like bacteria or viruses, you have to infect a cell, maybe even toxin, okay? And what is their function? I just said that. They what? They take this exogenous antigen, right? Break it down, display part of it, the epitope, whatever you want to call it, epitope in class what? Class MHC 2, yes, 2, okay? And of course they have to go ahead and do what? Find a T4 helper cell to be activated, all right, by the interleukin 2. Okay, interleukin 2. Sometimes though, they can function alone, okay, where they can be independent, okay, but oftentimes the discussion in most books go to T cell dependent antigens, but sometimes they can be independent and function on their own, but generally they are dependent on our T cells, but that's a little extra, but nonetheless. That. Okay, let's talk about this, antibody-mediated immunity. With this, what cells are we talking about immediately? No longer our T cells, but B cells, right? B cells. This is what we have an interest in, our B cells, right? Antibody-mediated immunity, or humoral immunity, if you will, which is the A. What cells? How do they get activated? We kind of just went through that a little bit ago, right? Okay. So with my drawing earlier, I had up there B cells. I had an arrow going down to plasma cells, right? Okay. And plasma cells would produce what? Antibodies. All right. Antibodies are the exact same molecular configuration as the B cell receptors that were on the original B cell up here. Okay? In my drawing here I said B cells, they have receptors on them, I called BCRs and B cell receptors. The antibodies produced by the plasma cells after proliferation and differentiation there, okay, are molecularly identical to the receptors that were on the original B cell, is only that they're called antibodies because they're in fluid. Okay, that's the difference. If you put them back on the B cell receptor, let's say we could do that, stick them on there, they would be B cell receptors. Okay, that's what they would be. They're only in fluid and they get a different name, they're called antibodies. All right? Same molecular configuration as the original, okay, or the mothership if you want. Okay, the progeny down here, all the same. Those are the same. So the difference is that receptors, okay, bind to the same antigen determiner epitope is just, I'm pointing down here like I have a picture. But now you have in the fluid, so they're called antibodies. Okay, so what do you know about antibodies so far? We did kind of a blood typing situation in lab. Okay, what did you notice as one function of antibodies based on that lab? They were able to what? Sometimes it worked really well. It didn't. They were agglutinate, yes. Antibodies now have a capacity to agglutinate or clump up. Right? That's one function of antibodies that you know of already. Okay? So we have kind of a list of some actions here, and we'll come to different types in a bit, but you already have an idea so far of what these antibodies can do. At least one function there is agglutinate. Agglutinate, all right? And we'll notice agglutination. We did notice that in the blood typing activity, but also antibodies are able to agglutinate certain toxins. Okay, like maybe you get bit by a snake venom injected there. Okay, you might want an antibody. Okay, that we're able to bind to our snake venom, agglutinate and clump it up. What does that do then for this? It's all agglutinated and clumped up. What does that make it more likely to happen? That it can be phagocytized and destroyed. So these antibody-antigen complexes, okay, they can be phagocytized more easily. Right? That's one action so far, okay, of our antibodies. Even though you may not know the types, and that's okay, you have an idea so far of at least one action, okay, agglutinating or clumping up. All right, and you notice that in the blood typing situation. Okay. Any other functions it can do?
Opsonization.
Yes, it certainly does. And what is that again?
Tagging or marking.
Tagging or marking a pathogen. Opsonization. And that also applied to what other group of plasma proteins, about 20 or so, they have the capacity, and once they're activated, which we don't do the cascade in this course, but they also have that capacity to opsonize. Again, that means tagging or binding to a pathogen. And if you're wondering again what that word refers to, kind of a weird deal, it actually means to make provisions for. All right? I like to ask that question, opsonized, where it butters them up. Okay, whatever it is, butters them up, makes them more likely to be phagocytized. That's what I mean by that. It has been opsonized. Complement has that capacity, as do antibodies. Okay, all right, so far so good. I think we're almost done here. At least, are we almost done? We got a minute left? Almost a minute, okay. So it does several other things, okay, as we'll see, antibodies do, and we'll get to not today anymore, but the different types of antibodies. And we'll see it also, they were able to neutralize, as we'll get to a little bit later, okay? And glutamate we did already, but also they are able to bind to mast cells and basophils to elicit histamine and heparin release too. So a little bit later there. So we're almost done with this chapter, and next time we meet, give me like 30 seconds for you to be out of here. Next time we meet is tomorrow, of course, and I will wrap this up, do a little bit of review, and then we'll continue with new stuff, which is respiratory, okay? Because this is big chapters. So please read the respiratory notes.